Hello, this presentation will discuss the rabies virus, including its structure, life cycle, transmission, and some of its various interactions. I will also discuss ongoing research aimed at finding an effective post-exposure vaccine for this virus. So what is the rabies virus? So it has a single-stranded, non-segmented, negative-stranded RNA genome. Rabies virus is a deadly but widely preventable disease, yet it causes almost 60,000 deaths annually due to encephalitis in areas such as Asia and Africa. Of note, these cases are typically in unvaccinated individuals and are usually caused by contact between humans and unvaccinated animals. There is obviously a higher proportion of wild animals that are unvaccinated than domestic animals, so the wild animals are typically the carriers and the ones to infect other animals and humans. The rabies virus has a bullet-shaped protein capsid, which is characteristic to the virus. The virus is typically around 180 nanometers long and 75 nanometers wide. The structure of the virus has two major components, which include a helical ribonucleoprotein core, RNP, which contains nucleic acid and a surrounding envelope. It also has a lipid-containing bilayer. There are about 400 trimeric spikes formed by the glycoprotein that are closely arranged on the surface of the virus. The virus genome encodes 5 proteins, which include nucleoprotein, phosphoprotein, matrix protein, glycoprotein, and polymerase. The structure of the rabies virus is determined by the RNA genome and the arrangement of these 5 proteins. The rabies virus infects nerve cells, or neurons, which are the smallest units of our nervous system. The central nervous system is made up of the brain and spinal cord, and the peripheral nervous system is made up of all the nerves that branch off of the spinal cord. The nervous system is responsible for all of the body's major functions. Nerve cells, or neurons, are chemoorganotrophs and work closely with astrocytes in order to perform critical functions. Neurons are heterotrophic, as are all human cells, and obtain carbon by consuming autotrophs and breaking them down via respiration to obtain cellular energy. Nerve cells perform catabolism using aerobic respiration, in which they break down glucose into ATP energy. During aerobic respiration within a neuron, the electron donor is glucose and the final electron acceptor is oxygen. Neurons are extremely sensitive to deficits in glucose and oxygen, and it is crucial that they receive large amounts of both at all times to avoid damage and reduce cellular metabolism. When the body is infected with the rabies virus via contact with an infected animal or individual, the virus attaches to a nerve cell and begins to replicate. This replication continues until rabies virus reaches the brain, which can cause it to swell and results ultimately in death. The rabies virus has many interactions with different cells and enzymes. The interactions that I will discuss today are those between the rabies virus and apoptosis, HLA-G expression, and the immune system evasion. Typically, apoptosis is the main defense mechanism used by the immune system to destroy cells that are infected with rabies virus and other neurotropic diseases. Apoptosis of cells infected with the virus is led by cytotoxic T cells and cytokines, including TNF-alpha, perforin, granzyme, and fast ligand. In order to continue progression through the nervous system, the rabies virus is able to upregulate AKT2, which suppresses the early apoptosis in neurons and allows the virus to continue to travel. This creates a high representation of the fast ligand, which in turn creates high apoptosis conditions in the immune cells. An interesting study of note suggests that there is an inverse correlation between the induction of apoptosis and the pathogenicity of the rabies virus strain. The HLA-G1 molecule is expressed by the neurons as a response to the appearance of the rabies virus in the body. The presence of the HLA-G molecule inhibits the ability of the natural killer and T cells to perform an inflammatory response in the presence of the virus. The HLA-G molecule is actually attached to the inhibitory receptors on the T cells and natural killer cells, which causes the destructions of these cell types. HLA-G is also capable of destructing the major histocompatibility complex 2, MHCII pathway, thus creating tolerance-mediating dendritic cells. The HLA-G expression can be triggered by cytokines and interferon, which are expressed when infection is present in cells and an immune response is needed to prevent further spread throughout the body. The progression of rabies virus infection is not deterred by the destruction of neurons that has been infected or by the immune response. Part of the genius of this virus is that it resides in the central nervous system, with immune effectors unable to cross the blood-brain barrier and clear the infection from the tissues. The virus is neuroinvasive, which results from a few different factors. Firstly, the virus avoids inducing neuronal cell death. Secondly, the T cells that enter the nervous system after infection are subjected to activation or apoptosis, which results from the overexpression of certain immunosubversive molecules that are in the infected nervous system. Those molecules include Fas ligand, HLA-G, and B7H1. The rabies virus is able to evade the immune system by preserving the neuronal network, as well as destroying T cells, allowing it to achieve neuroinvasion and transmission to the next host. A large population of people worldwide receive what is known as a PEP, or post-exposure prophylaxis treatment, which is typically very effective in preventing rabies disease onset if administered quickly after exposure. However, for those who have not received the PEP treatment and wait until symptoms of the virus occur, the disease is almost always fatal. Therefore, it is necessary to research and hopefully discover a potent and cost-effective treatment that can counteract rabies in patients who have already developed symptoms. In this study, 385 drugs previously approved by the FDA were tested in the search for an effective inhibitor of the rabies virus. One drug among these, pyrimethamine, was shown to inhibit the replication of rabies virus in low concentrations in vitro, as well as a synergistic relationship with ribavirin. The effects of this treatment were measured using an ELISA cat assay, which showed the ability of pyrimethamine to inhibit the rabies virus. Unfortunately, when tested in mice infected with the rabies virus, pyrimethamine did not prove to be an effective treatment. The results of the initial experiment involving pyrimethamine are depicted on the graph, which is attached here. This figure shows the relationship between the percent of inhibition, which is dose-dependent in this case, and the concentration of rabies virus within the body after treatment with pyrimethamine. The x-axis shows the concentration between 1 and 1,000 nanometers. The y-axis shows the percent of inhibition, which is evaluated 48 hours post-infection and is calculated using the ratio of absorbance value to the mean of the absorbance values that were previously obtained from cells that were infected but did not receive treatment. These values range from 0 to 100. The results of the graph show a positive correlation where the higher the concentration is, the higher the percent of inhibition. These results indicate that pyrimethamine is effective at reducing the concentration of rabies virus when used during in vitro experimentation. Unfortunately, during follow-up experiments, it was shown that pyrimethamine has no effect when used in vivo. This was made clear by experiments testing different concentrations of infection within mice over a 22-day period after infection. These findings indicate that, at this stage, this medication is not effective for use in vivo, only for use in vitro. Findings such as these lead to questions that would spark further investigation and experimentation. One of these questions is it the type of cell and cell structure specific to mice that make this drug regimen unsuccessful? Would pyrimethamine be more effective if it was used in a different type of mammal or even a human? Additionally, could this drug be effective on infected mice if it were used in conjunction with another medication? Answers to these types of questions would greatly help to make the progress towards a cost-effective and potent medication and reduce deaths due to rabies around the world. Although this experiment was not successful in trials with live subjects, its findings leave us one step closer to finding a solution in the future. An effective preventative vaccine has been created for this deadly virus, which has an astonishing mortality rate of 99.9% of those infected. Rabies virus can be detected both pre and post-mortem via antigen or tissue testing. However, it continues to affect our population, with around 60,000 rabies-related human deaths per year worldwide. Lastly, here are the sources that I used for this presentation. Thanks so much for watching.